Cells often undergo rapid changes in morphology, remodelling the actin cortex that underlies the plasma membrane during processes such as cell migration and cytokinesis. A striking example of cellular shape-shifting can be seen when cells are treated with microtubule depolymerizing drugs or placed in suspension. These conditions induce the formation of protrusions that oscillate periodically around the circumference of the cell, a phenomenon that captivates Maryna Kapustina from the University of North Carolina in Chapel Hill.
The beauty of this is that it's very stable. The cell could oscillate hours. It's really a very interesting phenomenon. And we do believe that this periodically protruding cell is really a great object to study some fundamental problem in cell biology.
Together with her colleagues Timothy Elston and Ken Jacobson, Kapustina found that the actomyosin cortex is highly dynamic in oscillating cells, polarizing at different parts of the cell periphery at different time points. The rounded protrusions generated by these cells have been thought to form by blebbing, where the plasma membrane detaches from the cortex and expands outwards before the cortex reassembles and retracts the membrane back into place.
We decided to really check if it's real blebbing or we still have a cortex underlying the membrane. So we co-express the membrane marker together with live actin, and we start monitoring the fluorescence signal when cell is oscillating.
Kapustina et al. Reasoned that if the periodic protrusions are blebs in which the membrane detaches from the cortex, the membrane and F-actin fluorescence signals wouldn't be correlated during protrusion growth and retraction. But the fluorescence signals actually remain strongly correlated in oscillating cells, indicating that the plasma membrane and cortex remain associated throughout the lifetime of a periodic protrusion.
When protrusion is growing, the signal is weakening and it became thinner. When the protrusion is coming back, it became always thicker and the intensity is higher. So it's highly correlated. It means that the transformation in membrane and cortex happens together.
The repetitiveness of periodic protrusions also suggests that they aren't membrane blebs, which tend to form more randomly.
So I was wondering how they start. So I plate cells and I detached them under the microscope. And it was a big surprise that oscillations start just immediately after rounding.
When cells round up, their surface area decreases dramatically in a short space of time, too fast for excess membrane to be removed by endocytosis. Instead, the plasma membrane, and presumably the underlying cortex, compacts itself by forming highly convoluted folds. The periodic protrusions seen in rounded cells could therefore arise from the cyclic compression and dilation of the plasma membrane cortical layer. Protrusions would form when the membrane and cortex unfolded, and then retract as the membrane cortex recompacted, an idea supported by measurements of cortical fluorescence intensity during protrusion formation.
So when you detach cells fast and the folds develop not maybe completely even, you have over compression on one side and maybe less compression on the other side, maybe this ignites this oscillation, but it's definitely possible only when you have excess membrane and cortex. You wouldn't see this on the fully spread cell.
The periodic folding and unfolding of the membrane cortex layer gives rise to a travelling wave of dense cortical actin around the cell periphery.
It's not easy to see on the confocal microscope, but sometimes you're lucky and this traveling wave goes exactly in the plane of the confocal volume. And it's important to understand that the trailing end of the wave is the densest part. And you can see often the very deep cleft where there's a lot of folding and compressed material. And when the compression becomes too much, the unfolding happens and the protrusion is developing from the end of the wave. It's like something telling cells, okay, I can't hold anymore here. Let it go.
One molecule that regulates the folding and unfolding of the cortical layer is myosin II. Treatment with the myosin inhibitor blebistatin redistributed actin evenly around the cell cortex and abolished periodic protrusions, whereas inactivating blebistatin with blue light restored the oscillatory phenotype. As a way to store and release membrane and cortex, compression and dilation of the cell periphery may be involved in other, more physiological shape changes, such as those that occur during cell migration. Kapustina et al. Found that oscillating cells are motile and can even migrate directionally towards a chemoattractant. Indeed, periodically protruding cells look remarkably similar to cells undergoing amoeboid-like migration through 3D environments, a mode of motility previously thought to involve membrane blebbing, but that may instead utilize cortical folding and unfolding.
And if it's the case, we think periodically protruding cells will be a great model to study this 3D amoeboid type motility, because you can do it basically on the almost 2D. So, of course, we're going to do more experiments and see how it works in more details. We want really to look what other actin-binding protein is involved, because to hold all this structure, to hold these folds, it's probably not only the myosin involved, it's a lot of other actin-binding proteins. And we already have some preliminary results that phosphoinositide play maybe major role in this phenomenon.
Kapustina and colleagues also want to model the mechanics of periodic protrusions, but in the meantime, you can learn more about how compression and dilation of the membrane cortex layer generates rapid cell shape changes in the paper by Kapustina et al., published in the January 7, 2013 issue of the Journal of Cell Biology.